In this lesson, we're going to explore the physical changes that occur during pregnancy, from conception to birth, from different perspectives. This lesson corresponds to Chapter 3 of your textbook on prenatal development and birth. First, I'm going to discuss the process of conception and the transfer of genes from the parents to the developing fetus. Then I will discuss the different phases of prenatal development from the germinal stage to the embryonic stage. And finally, I will discuss the changes that a woman will notice in her body during each of the pregnancy trimesters. I will also include some useful videos throughout the presentation so that you can visually understand how an infant develops. The first step in development happens at the point of conception, when we receive a combination of genes from our mother and father that will shape our experiences throughout the rest of our lives. Our genes, which contain all of our DNA or genetic material, are packed into 23 pairs of chromosomes, or 46 individual autosomes. At the point of fertilization, the 23 chromosomes of the sperm combine with the 23 chromosomes of the ovum to form a new organism called a zygote. The zygote now has its own unique combination of 23 pairs of chromosomes that represent their unique genetic code. The 23rd pair of our chromosomes is the sex chromosome. The mother always contributes an X sex chromosome, so the actual sex of a child is determined by the father. In men, about 50% of their sperm carry an X sex chromosome and 50% carry a Y sex chromosome. Therefore, if a male sperm delivers an X, the zygote will end up being a girl. In contrast, if the male's sperm ends up contributing a Y, the zygote will end up being a boy. In most cases, humans are conceived and born one at a time. However, roughly 3.3 out of every 100 births in Canada are multiples, which typically means twins. Now, there are two types of twins: monozygotic and dizygotic. Dizygotic twins are also referred to as fraternal, and they represent about 2/3 of all twins born. Fraternal twins are conceived when two eggs are fertilized by two separate sperm. Identical twins, or monozygotic twins, are conceived when a single fertilized egg separates into two parts, and each part develops into a separate individual. Interestingly, the rates of multiple births has actually tripled in Canada since the 1980s. Part of the reason for this increase in multiple births is the fact that there are more and more women who are becoming parents after the age of 35. The research suggests that increased maternal age is associated with an increase in the natural conception of multiple births. In addition, an increase in maternal age is also associated with the increased use of assisted human reproductive techniques. And we know that multiple births are more common with women who are using artificial reproductive techniques such as in vitro fertilization, because during this process, many embryos are implanted into the uterus to increase the chances or probability of a pregnancy. Prenatal development, or gestation, refers to the process that transforms a zygote into a newborn. This entire process lasts approximately 9 months. The first stage of prenatal development is called the germinal stage, and this begins at the point of conception and lasts for about 2 weeks into the pregnancy. By the end of the second week, during this stage, the zygote travels down the fallopian tube and ends up implanting on the uterine wall. At the point of implantation, the zygote undergoes rapid cell division, and each cell will end up becoming a specialized part of the organ system of that infant. During this stage in particular, the organs that are developing are the placenta and the umbilical cord. The placenta is the organ that allows oxygen and nutrients to flow from the mother to the fetus via the bloodstream, and the umbilical cord is what connects the embryo to the placenta. Also during this stage, the amniotic fluid sac forms. This is the sac in which the fetus floats until just before it is ready to be born. The second stage of prenatal development is called the embryonic stage. This stage begins at the end of the second week of pregnancy and lasts until about week 8. During this stage, the neurons, which are specialized cells of the nervous system, begin developing to form the major organs and systems of the infant. This process is called organogenesis. So at this point, basic parts of the organs are beginning to develop, including the limb buds, the eyes, the heart, and other organs such as the ovaries in females and testes in males. At the end of this stage, the embryo is approximately 2.5 centimeters, and the embryo contains enough features that it can actually be recognized as a human embryo. Also during this stage, the embryo is very vulnerable because this is when all the major organs and physiological structures are being formed. The third and final stage of prenatal development is called the fetal stage. This stage starts at the end of week 8 and continues until birth, so lasting about 7 months. During this stage, maturation and major organization of the organs and nervous system are occurring, especially those organs that are essential to survival outside of the womb. So this includes the lungs and the brain. By the end of this stage, the fetus starts to inhale the amniotic fluid as a means to develop their lungs and to practice breathing to prepare themselves for birth. They also, by the end of this stage, will start to rotate to an upside down position to prepare for birth. Now we're going to look at pregnancy from the mother's perspective. Customarily, pregnancy is divided into 3 trimesters, including 3 periods of approximately 3 months each. In the first trimester, or the initial phase of pregnancy, the zygote implants itself in the lining of the woman's uterus. The hormonal changes that are triggered by the implantation of the zygote produce physiological changes in the mother's body and the characteristic symptoms of nausea or morning sickness and fatigue. At this point, the uterine lining begins to thicken to support the developing embryo, and the uterus enlarges and shifts, which puts pressure on the mother's bladder. During the first trimester, prenatal care is particularly important for both mother and baby to prevent birth defects. Recall that most of the baby's major organs develop during the first 8 weeks of pregnancy. During the first trimester, the mother is also at the greatest risk for miscarriage as well, which is also why prenatal care is so essential. You can see a summary of the different phases of pregnancy in Table 3.1 on page 59 of your textbook. The second trimester of pregnancy lasts from about week 12 to week 24. During this stage, morning sickness typically goes away, and a pregnant woman may have an increase in appetite, which helps her to gain weight as the uterus expands. This is a particularly exciting time for most pregnant women because they start to feel the baby's movements for the first time by about the 16th week. The baby's gender can also be determined by about week 13 by doing an ultrasound. third and final trimester of pregnancy begins at about 25 weeks and continues until the mother goes into labor. During the third trimester, a pregnant woman gains more weight and experiences greater abdominal enlargement and consequently more discomfort. However, the women begin to feel more emotionally connected to the fetus as they prepare for birth. In preparation for nursing, a woman's breast begins to secrete colostrum during this trimester. Colostrum is a substance that is low in fat and high in carbohydrates, protein, and antibodies, which helps keep a baby healthy. Prenatal development is not immune to internal or external influences. Things can go wrong. Approximately 2 to 3% of babies are born with congenital abnormalities or birth defects. Most of these abnormalities are very rare, some are preventable, and some need not have permanent consequences for the child. Some of these congenital abnormalities can be caused by genetic defects, and others are caused by chromosomal errors. For example, the most common chromosomal error is a trisomy 21, or Down syndrome, in which a child is born with 3 copies of chromosome number 21. The risk of bearing a child with trisomy 21 or Down syndrome increases with maternal age. Some birth defects, however, can be caused by maternal illness during pregnancy or exposures to teratogens during prenatal development, the latter of which is mostly preventable. Teratogens are agents that cause damage to an embryo or fetus and include things such as drugs and/or alcohol. The damage caused by exposure to teratogens depends on the intensity and duration of the exposure, as well as the developmental period during which the exposure occurs. The greatest risk from exposure to teratogens occurs during the first 8 weeks of gestation, because this is when most of the embryo's organs are starting to develop. For example, mothers who are heavy drinkers are at increased risk of delivering infants with fetal alcohol syndrome, or FAS. Infants born with fetal alcohol syndrome are generally smaller and have smaller brains. They also tend to have organ abnormalities, including heart defects and hearing loss. Children with FAS, or fetal alcohol syndrome, also have distinct facial characteristics, which include small, wide-set eyes, a flattened nose, a thin upper lip, and most children with fetal alcohol syndrome end up with learning, behavioral, and/or cognitive deficits.